I'm going to talk about advances in in vitro endpoints for inhaled drug delivery assessment today. And just to kind of put this in context, well, where does this fit in the pipeline? So we're really focused kind of on preclinical. So we've got a couple of candidates and really want to get them through, really understand how that formulation behaves in a whole system. How do we de-risk that before we go into humans? And we can reuse in vitro quite a lot on this pipeline. And preclinical is where kind of in vitro is emerging. It has been emerging really for the last kind of 10, 15 years. But there's been quite a lot of advances recently where we can really accelerate this. And you've probably seen in the news over the last kind of few years, the FDA released something a couple of years ago about having a real belief in NAMs and how that can replace animal needs. And the UK have responded. They responded in November this year. There's a paper about replacing animals in science and a real strategy behind that. But it's kind of a milestone paper for the UK. And we've been in a lot of consultation with them, this being a NAMs-driven company. And I guess one of my responses is we do have many of the tools already there, and the innovations are there, and they're kind of waiting to be used. But what's lacking is probably that framework, that full understanding of how we can integrate them better and into the framework, the regulatory framework that needs to be. So I think the future is strong for NAMs, for different approaches, but there are absolutely some challenges alongside that too. So what kind of preclinical questions are there, and where are we using in vitro approaches in terms of safety? So pharmacology, target identification, and does it do the job? Product performance, which is a lot of people in the audience where you're focused on, is what's the aerosol like? How does it work? What's the performance of that product? How does it deposit when the lungs? What's the delivery like? And then we kind of move on more to the biological areas, so the pharmacokinetics and also that toxicology. Before we look at excipient, device component safety, and translational, how does that translate into a human? And the inhalation is a lot more complicated than other areas, because we do have that device, that excipient, the exposure, conditions of deposition. But where I'm going to focus on today is toxicology, and should we be integrating this earlier into our decisions? Could we take some of the toxicology information that will help us get things more rapidly through that preclinical stage? So inhalation, toxicology in inhalation comes into two phases. There's inhalation toxicology, but there's also safety pharmacology as well. So the inhalation toxicology is really about what happens in that respiratory tract following either systemic or direct exposure. So there's some basic questions that we can answer there. So what locally happens in that tract? How are the epithelial cells in particular being affected? What does the dose delivery look like? What does the exposure? How does that exposure change how those cells behave? And then some kind of off targets. So what happens via systemic exposure from the inhalation route? How does it affect the other organs? What happens when we take those things quite regularly? And what's often overlooked is that immunotoxicity. We don't just have a barrier cell in the lung. There are many other different types of cell, and particularly immune cells have a very important response in how our lungs react. And in terms of local respiratory tract toxicity, we've got some really quite advanced models that have been used kind of for the last 20 years. Some of these are kind of going through ring trials now, which will probably appear kind of in OECD guidelines possibly in the next kind of 6 to 8 years. So things like looking at cytotoxicity, they're very kind of black and white answers in toxicity. Is it dead? Is it alive? Has the barrier function been removed, or is it still functional? How stressed is that cell? Is there any DNA damage? And those things are quite easy to assess. But what do they actually mean when we translate them into humans and away from that dish? And the other aspect of safety and toxicology is safety pharmacology. And this is a much bigger question, not necessarily on the cellular level, but what actually happens to the lungs. And when we're looking at in experiments we're trying to replace in vivo, it's things like respiratory rate, tidal volume, how many breaths you take in a minute. And that's all very easy endpoints to get from a rat, and we can then understand actually is there any broad issues. But they don't translate to a cell. My cells in a dish don't cough, they don't breathe. So actually replacing these kind of endpoints can be quite challenging. But in the last kind of 5-10 years, there are lots of other surrogate in vitro endpoints that we can use. So for bronchoconstriction, we can look at airway smooth muscle. For cough and irritation, there's specific activation of receptors. For inflammation, the macrophage is really, really important in terms of some of those targets. And also looking at off-target respiratory effects via other ions. So whilst we can't directly look at those effects, we can look at the pathway and the mechanism of that, and really go back and understand that in an in vitro culture. And the approaches to more complex safety questions, they're not straightforward. It's a continual process, redefining and defining the question again and again, getting that appropriate level of complexity in the model. Epithelial cells are absolutely great for looking at epithelial responses, but that's not the only thing that's happening in the cell. And really, we're moving towards more of a mechanism-driven adverse outcome pathway where we understand each and every individual component of that and try to answer those questions. So thinking about the question, are we picking the right model just because it's a complex model that really looks like the lung? Is it the best model to use? Thinking about the exposure, what kind of endpoints we do and how we interpret it. And that circle goes round and round. So what is available at the moment? So in terms of mechanism-driven safety, there are models with just epithelial cells, there are co-culture models, there are models just with immune cells on their own, but it's picking the right model and the right exposure. So things that are quite straightforward, things like cell morphology and cell health, understanding that dead or alive response. But in terms of the mechanism, there's more and more emerging technologies now that we can use, looking at inflammation, but not just whether a cytokine is present, but whether a pathway is particularly activated. And there's new models coming on for sensitization, which is something that's very hard to mimic in an animal model, but actually we can do that in a human in vitro system. Questions like clearance, how long is the medicine retained there? How is it affecting cell uptake and motility? Absorption, disease-driven things, secretory function, and that translation. These are just kind of the tip of the iceberg, but the ones that are established. But there are more and more things available now other than, is it just alive or dead? And what I'm going to concentrate on is, for just the last kind of 6 minutes, is thinking about the immune and the inflammation response, and particularly looking at morphology and some of the functional aspects that we can do to answer specific questions in the lung. So in terms of toxicity, whilst epithelial cells are obviously really important, alveolar macrophages are actually probably arguably the most important cell in the lower airways. So when you're getting particles right down to your alveolus, alveolar macrophages are normally that first responder, and understanding how they respond first is often probably the best way you can understand the fate and the safety of a medicine in the lungs. So about 10, 12 years ago now, there was a big problem when small molecules in the lung, and looking at how we can understand how changes in macrophages that we were seeing in rats, actually, are they effective to humans? Just because you see an enlarged macrophage that looks quite angry in a rat, does that mean what's going to be happening in the human? And actually, is it relevant? What does it mean for diversity? And actually, those questions from 10, 12 years ago are still with us now. Maybe not with the same kind of molecules, but the same questions are coming up in off-target toxicity with things like ADCs, and also other inhaled poorly soluble molecules from the atmosphere. So understanding that whether things are adapt or reverse, that's kind of where in vitro is really going to. We're very good at saying, yes, it's alive or dead, but actually, if we're getting changes, we expect changes when we breathe particles in, but how significant are those changes to cell health? So the technique I'm going to talk about is kind of high content imaging. So that's a technique that has the direct endpoint as you would in an in vivo test. So in an in vivo assessment, you would have slices of the lung, and it'd be looked at by histopathology, and the cell would be graded in terms of how healthy, how large and foam enlarged it was. And we can do that same process on a very individual cell level with a human or a rat macrophage in a dish. So it'd be grown in a dish, exposed to a cell culture, stained with a variety of cocktails, and then analysed for specific features. So the cell kind of features that we've kind of worked over the last kind of 10, 12 years are cell health, its activity, the area of the cell, and the area of its nucleus, and how round it is, very important to how it's responding, and also those vacuolation features. So the picture here, you can see some very clearly defined vacuoles, and those processes will change with different types of exposures. So how can we use that? So first of all, to really understand interspecies differences, the rat has been used quite historically for looking at inhalation safety, but the rat immune system, the way the rat breathes, in fact, the entire physiology of the rat lung respiratory system is incredibly different to humans, and that difference also is quite notable on the immune sense as well. But we can look at detailed interspecies characterization. One of the biggest problems is size in terms of the macrophage. The macrophage is about 3.5 times bigger in a human model than they are in a rat model. So when you're thinking about lung overload and particulates, the rat model just doesn't represent human. They cannot take up as much material. So we have done over the past lots of very rat and human alveolar macrophage monitoring to understand, well, what's the baseline like? Can we transfer data from human changes in macrophages to rats and vice versa? And we can get quite a steady baseline of those. And most, we've been looking at cell health area, right down to kind of membrane permeability, how the size of those areas of the cells is, how many lipids are inside those cells. And we've done quite a lot of maths and worked with histologists over the years to find out, well, actually, do we need to report on all of this? Does it have to be this complicated? And we've actually brought it down to just kind of 4 main areas as a really first kind of tier 1 screen. And that would be the activity of the cell, the area of the cell, the number of vacuoles that cell has, and how much of that surface the vacuoles are taken up. And with those 4 categorizations, we can overlap them, so we can have cells that are large that have many vacuoles, or cells that are just large on their own, or cells that are large with a higher activity. And we can create this phenotype or fingerprint, which is at the bottom here. And that fingerprint is really, really representative every time we do it. So we've got a lot of historic data that we can characterize. This is what a healthy cell looks like, which is a really good starting point. Establishing the baseline is often half the problem. We can then look at, over the past few years, we've put many, many compounds through these to try and understand, well, how does the cell respond to different compounds? So this is an example of bronchodilators. The bottom is the human. You can see very small responses. In the top, you can see the rat, and the rat has quite marked different responses, but they are a class effect to that molecule. And we can then understand, well, a species acting the same. We can then explore the phenotypes in different areas as well. So looking at different types of drugs that are inhaled, so things like salmeterol, fluticasone, they're on the market, they're safe, but everything does have a response. But where does that response become adaptive? So really establishing a baseline. I mean, amiodarone is something that is on the market that causes off-target toxicity. And you can compare this to other things. So this is a molecule, so it's a silica molecule that has a very, very different phenotype. Obviously, if you breathe in silica, it's not particularly good for your lungs. And you can see that signature fingerprint changes. And then the last bit I was going to touch on was alveolar motility. So not just looking at the morphology, but also function. So function can tell us quite a lot about toxicity and how motile those cells are. So this is an example of salmeterol, and this with different concentrations. And the cells are generally have the same motility over time. Fluticasone is slightly more poorly soluble, but again, it has minor changes in motility, but generally that motility doesn't change. And then amiodarone is something that would be classed now as toxic. You can see that the initial stress response is quite high. The cells are very, very motile, up to kind of about 7 hours. And when you get to a more toxic concentration, those cells then don't respond. That's the lower line. So looking at really sensitive things like motility actually can tell us in advance of morphology changes where a product becomes adverse. And I get the future is probably profiling multiple endpoints. There isn't going to be one in vitro assay that's going to replace a whole kind of IND study in a rat. That's not realistically possible. But if we can start to build up databases of multiple endpoints with key toxicology questions, that's kind of where the future sits for in vitro toxicity in the lung. So in summary, I think the tools exist. They're there. We probably haven't applied them in quite the right way that we need to at the moment. Thinking about the framework and the context of use that we apply those the assessments in is really important. And getting a testing framework, the wheels are in motion for that, for there to be a rule book on how to do it, but we're absolutely not there yet. I'd just like to thank staff at ME1. We had a collaboration with FIO, who put the motility work together with as well. And some of this work that started actually from a Crack It Challenge consortium way back in 2014. Thank you for listening.
Thank you. Any questions for Vicky? While people are thinking, can I just ask Vicky?
Of course you can.
You mentioned NAMs a few times in your opening slides. Can you remind us what they are?
Of course I can. Sorry, I forgot my audience. So NAMs is new approach methodologies. So it's a term that's been probably coined now for the last 5 years or so. So it's in vitro. Or it could be in silico. But it's new approaches rather than just using animals as a endpoint. So there's a lot of strategies around Europe, but the UK have just released their statement on it as well.
Okay, and if there's one further question from me then is, in your suite of endpoints for macrophages, there's not a phagocytosis endpoint. But I
think it was on there.
It
was in the small, yeah. So phagocytosis is important. I think the engulfment and the mechanism of engulfment, because there's not just phagocytosis, there's many different types of ways of engulfment. It's part of understanding that mechanistic approach. And I don't think there is one. I don't think you can take a chemical through a standard set, because it very much depends on that pharmacology of that molecule. But yeah, engulfment is very important, understanding where it's located within the cell.
Thank you very much.
Thank you.